What's up everybody? So in this video we're going to be talking about everything that you need to know regarding transcription and translation. So picture this, we have here Bob the Builder, right? And we know he loves to build things, so he comes up with this crazy idea to build a rocket. Now, he goes to the top floor of his house because there he has this instruction manual, this book. Now this book doesn't only have the instructions to build a rocket, but it has the instructions to build many other things on Earth, right? It's like an instruction book. So he doesn't need the entire book, he only needs a single page. But so as he tries to go in the room, he realizes he can't because his dad is there working and his dad doesn't want him to go in. So he asks his dad, "Dad, can you please rip out this one page from this book because I need it to build my rocket?" And his dad's like, "Wow, no way. I'm not going to rip out a page from this brand new book. But what I will do is we'll take it to the photocopy machine and there I will copy the page that you need and give it to you. That way the book is fine and not destroyed." So that's what happens. So the dad photocopies it, slides this page under the door, and now Bob the Builder has this instruction manual to build this rocket. So now he heads down to the garage. Why? Because the garage has the materials, right? That has the materials that he can use now to build this rocket. So ultimately he has three things. He has this instruction manual, and he has the materials, and he has himself, because he is like the tools. He is going to build this rocket. He's going to use his tools and things like that to build this rocket. So why did I tell you this story? Because this story is a perfect analogy of transcription and translation, and you'll see exactly how. So how? Why did I just tell you the story? So one of the most important molecules in our body is called proteins. Proteins. They are made up of amino acids. It's basically, proteins are a long chain of amino acids. Now we have around 20 different types of amino acids in our body. Now depending on the order that we put these amino acids, that will give us a different protein. And also depending on how long we decide to make this protein, that will also influence the type of protein we have. So we can have many types of proteins depending on the structure we give them. Now, how do we make a protein? A protein is made by transcription and translation. So a protein, pretty much in my story, my analogy here, is represented by this rocket. Transcription and translation is a process of trying to make this protein, this final boss product that we need to be able to do many things in our body. Because proteins can have many functions, like acting as an enzyme, helping you build muscle. There's unlimited array of functions that a protein can have. So let's start getting into a little bit more detail. So here we have Bear Grylls. And as I tell this story of transcription and translation, I will constantly refer back to this analogy so you can see exactly how this analogy is just so perfect. And anytime you forget what transcription and translation is, and you think of this story, it will help you a lot. So here we have, what do you call him? We call him Bear Grylls, right? Bear Grylls, we know he loves to go out and explore and do all these kinds of things, right? He's an adventurer. So let's say one day he goes out and hurts his hand and now he's bleeding real bad. So what needs to happen is, right, his body is going to want to stop the bleeding so that he cannot lose any more blood. Now to do so, his body is going to have to make a protein that's specific for stopping bleeding, a protein that is specific to clot blood so he stops bleeding, because you're not supposed to bleed for that long, right? So after you cut your hand, you'll stop bleeding within like 10-15 minutes. You're not supposed to bleed for days, right? So what helps this is some proteins that help clot the blood. Now, how are these proteins made? So basically, in his hand, he's going to have some cells, right? Because we know humans are made up of trillions of cells. Now, the cells of his body will make this protein. How? So step one is we got to go to this nucleus. So this is our beautiful cell, and we're going to care a lot about the nucleus. Why? Because the nucleus serves as the instruction manual. This is the code that codes for everything, all the proteins in our body. Now, how does that relate to our Bob the Builder story? So we know for our Bob the Builder story, Bob the Builder went to the library, which is represented by the nucleus here, because the nucleus has all the DNA, like the library had this book. Now, remember, as Bob the Builder went, he didn't want the entire book. Like, he didn't want the entire book. Just that way, we don't want the entire DNA. Our DNA in one single cell, in one single nucleus, is 6 meters long. But to make this specific protein, we only need a small part of it. So the first part of this whole process of transcription and translation is finding the specific piece of the DNA that we need to code for this protein. So that's what happens. We find it, and guess what? Just like in Bob the Builder, we copied that specific piece of the instruction to make that rocket. We're also going to copy just one piece of the entire DNA into another molecule, which is basically behaving like a piece of paper that is specific to this coding for this specific protein. Now, to copy the page from the book in Bob the Builder story, we needed a photocopy machine. Now, there's a molecule which we'll get to later, which behaves like a photocopy machine, which is going to do exactly that in our cells. Now, this process that I just explained is called transcription. The process of copying the specific piece of DNA into an mRNA. Okay, so basically this process, this is called transcription, and we'll get into the actual process a bit later. Now, this happens in the nucleus. So transcription happens in the nucleus. Just like he turned his book, he copied the specific page of his book into a single page in the library, right? Not outside the library, in the library. So now what happens next? Next, we're gonna basically, normally DNA is too big to be able to, DNA is not allowed to leave the nucleus. But because we copied now this DNA into the specific piece we needed, and this is not DNA, this is something called RNA, and we'll get to this a bit later. RNA, unlike DNA, can leave the nucleus inside into the cytoplasm. Why is this important? This is important because the cytoplasm holds all of the materials and tools to be able to build this protein. It is not in the nucleus. All the materials and tools are in the cytoplasm. So it's very important that we send out these instructions into the cytoplasm for that reason. So now what's going to happen is, remember in the Bob the Builder story, this paper now combined with some materials and tools, which is him, to make this final protein. So the same way in our bodies, we're going to combine this instruction piece of paper here that codes for this protein specifically to be able to combine it with both tools, which is this, and tools, which is this one, and materials, which is this one, to make the final protein. And don't worry about what these are now. That's the whole purpose of the video. We're going to go into some detail. So this part right here, the second part, converting this paper here, this paper into the protein, or this code here specific for this protein into the protein using some materials and materials and tools, is called translation. So that's the second part of this process. And this one happens in the cytoplasm, because that is where all of these tools and materials are. So this right here was a quick outline of transcription and translation, and you can see I related it to the analogy that I made. So anytime you forget, you can think of this and think, oh yeah, I remember now. Okay, it's not that useful, but I found it really useful for me in case I forgot, because you have to remember so many things in the IB. So now, let's zoom in. Let's go into some more details on each of this. So we're going to go now first into transcription. Now remember, transcription happens where? Inside the nucleus. So we're going to zoom in here into the nucleus. That's where we're going. So now here we're zoomed into the nucleus. You see here the nucleus. This darker purpley part's the nucleus. Right outside is the cytoplasm, right? I can show you again. We have the nucleus here, and right outside is the cytoplasm. So, like I said before, just to reiterate, our DNA is way too long, right? And it codes for many, many, many different types of proteins that have different functions. But in our example now, we are saying we need a protein that helps Bear Grylls bleeding to stop. So now, we don't want the entire DNA, we just want that one segment that codes for that protein. So now what's going to happen is, a molecule called RNA polymerase, which looks like this, which I showed it to you on the previous page. This molecule, or this enzyme rather, because it ends in ase, is going to have very important roles in transcription. So first of all, DNA is normally coiled, right, like this. Now this makes DNA very, very, very difficult to access because it's all coiled up. It's like it's hidden inside. All of the code is hidden inside. So what we need to do is this molecule needs to unwind the section of DNA that we care about. Okay, the section that's going to help code for this specific protein. So that's what it does. It unwinds it. It unwinds this DNA. Now we can see it more clearly. Now remember, the code we care about is this one right here. This one right here. We know DNA is double-stranded, so we got this strand here, and we also have this strand here. Now each strand has a sequence of letters, so we can see here blue, red, or blue, red, green, red, blue, blue, green, red, right? This is one code going down there, and this is another code going down here. So we don't care about both of these, we care about one of these. So we only want this code right here. So we don't want both strands. We don't want to copy both strands. We want to copy one strand. That's why when I showed you up here how we copy the DNA into one strand, that's because we only care about one code, not two codes. DNA is double-stranded. So that means that there's two strands with two different codes. We only care about this one. Now, like I said, DNA is not allowed to leave the nucleus into the cytoplasm. So we need to copy this code into another form called RNA, which is very, very similar to DNA. Once we copy it into that form is in fact allowed to leave into the cytoplasm for the next step. Okay, so that's some basics. Now that you know that we care about this specific code, we need this enzyme to do one more thing. Not only does it unwind the DNA like I just showed you, it also goes in and slices open these bonds. You know, do you see these bonds holding the two strands together? It slices it open so that the two strands can be separated, because then we can have complete access to this specific strand that we really care about. So that's what happens here. Let me show you. So now we cut open these strands, and the two strands are separated. Now remember, this is just a quick recap for you in case you forgot a little bit. Remember, DNA is made up of these four letters: A, T, C, and G, right? That's the code. DNA is made up of these four letters put in different orders and all these kinds of things. So we know when we come to this image, right, this here is our code. So these are different letters coding for something, coding for this protein that we want to make. Now, like I said, now we're going to copy this code into an mRNA. So it's not a DNA, because DNA is not allowed to leave, and DNA always has to be double-stranded. But we don't want two strands, we only want this one strand. So the only way we can copy this DNA into one strand is by making it into an RNA, because RNAs can exist as one strand. So now let's quickly label some important things. So this strand here that we want to copy, we give it a name. We call it antisense strand. Now the opposite strand that was attached to this antisense strand, we don't care about, and we call this one the sense strand, Then we have two more other names: the promoter region and the terminator region. So the promoter region is the place where our gene, where the gene starts that we want to copy. So this is the entire gene we want to copy. Now this is going to be the start site of where we start copying it. And this will be the end site, the terminator region. So promoter region just means the starting region, and this means the ending region. Because we don't want to continue copying the whole DNA, we only want to start somewhere and end somewhere, right? So that's why we need this promoter region and terminator region. Now, one last quick thing. Remember from DNA structure that DNA, right, is made up of these two strands. Now each strand has a top and a bottom, which we call a 5' end and a 3' end. So let's consider this darker strand here. Say this is its 5' end. Now if you follow it along, this will be, the other side will be considered what? The 3' end. Now we know that the other strand will work the exact opposite way, right? They run anti-parallel. They are the opposite. So instead of this side being 5' end, it's going to be what? It's going to be 3' end. And instead of this side being 3' end, it's going to be 5' end. So they work the opposite. And why is it like this? If you don't know, I'm not going to explain now, but I explained it in my DNA structure video. So I really, really recommend you check that one out before you even continue watching this video, just so things make a little bit more sense for you. So now let's get started a bit more. So now we're going to have this very important molecule come in, right? This molecule, remember it. This was what? What was it called? RNA polymerase. Thus far, it had very important roles, right? It had the role of unwinding and unzipping this DNA so that we can have clear access to this antisense strand. So what it's going to do first is it's going to go to this promoter region where it's going to start copying this specific antisense strand into an mRNA molecule. mRNA molecule. So let's say it starts. So here it starts. I'm going to try and get this right. So now it's going to grab, I'm going to move it out of the way just for the sake of, so it doesn't look so messy. So I'm just going to move it here. But just know it's supposed to be over here, overwhelming and doing everything here. So what this enzyme is going to do is very easy if you think about the name. RNA polymerase. What does polymer mean? Polymer. A polymer means a chain, a long chain of something. ACE means an enzyme. So this is an enzyme that's going to make a long chain of something, a polymer of something. A polymer of what? A polymer of RNA. So it's going to make a polymer of RNA using this antisense strand as a template, as an example, right? It's like coming back here to the DNA. Remember, we copied this book, but we used this one of the pages as an example of what to copy, right? So that's the same thing in our DNA. Okay, so basically there's these free-floating nucleotides, RNA nucleotides, remember? RNA, so I changed the color so it's red to represent RNA. RNA is slightly different from DNA, remember? RNA, instead of being A, T, C, and G, is very similar, but one difference, it has A, C, U, and G. So instead of having a T, it has a U. And RNA can only be single-stranded, not double-stranded. But that's why RNA is so useful in this example, because we only want to copy one strand. We don't want to copy both strands of DNA. So that's why RNA is really, really useful for us. So this starts happening. I'll try and skip a little bit to make it quicker. So it starts copying one by one, doing what you call complementary base pairing, because we know A always binds with T and so on, right? So we start copying. Give me a second, I'm just trying to make this smoother for you. So we start copying, he starts copying in this direction from the promoter region this way. I skipped this X section from now, but in reality it starts all the way from here and it goes all the way down. I'm just filling in from here, but bear in mind I should have filled stuff in from here as well. So anyways, it copies down this direction, and essentially what we're forming here is an mRNA. And right now maybe you're a bit confused. Maybe you're looking at this and saying, okay, we're working on this antisense strand, but what we're forming is actually the exact opposite of this antisense strand, right? Because we're doing complementary base pairing. So what we're forming here, with this mRNA we're forming, is actually very similar to the sense strand, not actually the antisense strand, right? That's true. Look here, we have the orange, the blue, the blue, the green, the green. The only thing that's different is mRNA doesn't have T, so it doesn't have this red molecule that DNA does. Instead, it has this blue molecule. Okay, so you're right in thinking this. You're thinking, okay, I thought we're copying this DNA. You'll see exactly later on in this video how we are in fact using this as the code, even though right now it seems like we're copying something different. You are right in thinking this, but you'll see later on in this video why this makes perfect sense. So anyways, this continues, and this RNA polymerase goes all the way down and finishes this gene, copying the entire antisense strand into what we call an mRNA molecule. So let's pretend it started down here, he copied it all the way, and as soon as he finished copying it, it will start detaching. So the bonds in between these nucleotides here are very weak, so they don't stay together. So after it is synthesized, it will leave the nucleus through the cytoplasm, as you can see here. And then the sense and antisense strand of DNA will basically bind back together and the DNA will recoil. This way, the DNA was unaffected, right? It was completely unaffected. And going back to the story again, remember, when we photocopy this book, do we damage it? No, we keep it exactly the same. So it's the same with the DNA. By copying a piece of the DNA, we don't affect the DNA. It remains unaffected. It remains in the same condition it was before we copied anything. So anyways, now we have this mRNA. And by the way, we copied it. We synthesized this mRNA molecule from 5' end to what end? Give me a second. To a 3' end. right? Because this strand right here, the antisense strand, goes from 5' end this side to 3' end this side, which means this mRNA will be the reverse. So from 3' end to the 5' end. So that is it. That is really all you need to know for transcription. So we just covered this part. Copying this piece of DNA into an mRNA molecule. mRNA molecule. So now, this mRNA molecule is called an mRNA molecule, which means messenger RNA. It's going to serve as the code, the message to make this final protein, this message between the nucleus and the cytoplasm. So messenger RNA. So here we go now. So now we have here the same kind of picture, just we shifted it a bit. So the nucleus is up here now, and now we're in the cytoplasm. Here is our what? What is this? The mRNA molecule that we just synthesized. So remember, I'm going to quickly explain some things about mRNA that you should know. So mRNA is a code, right? It's simply a code. Now, when you read an English book, how do you understand the book? Because you read the book word by word. Each word means something, right? That's the same thing here for mRNA. So when you read this mRNA, you read it word for word. So in mRNA, the words are in letters of 3. So AUG, these 3 letters is one word, and we call that a codon, a codon. So the next 3 letters, CAU, that's another word. So that one means something else. UGG is another word, and that one means something else. So unlike in English, how we have words of different lengths, in mRNA, words are always 3 letters. So one end of this mRNA we call the start end, and the other end we call the stop end, because we want to basically read this entire mRNA and make the protein, right? We don't want to start from the middle, anything like that. We want to read from the start to the end. So the start area is called the start codon. AUG is always the code. AUG is basically the word that represents start. AUG, always, AUG. And I remember it as AUG stands for August. And that's just how I remember it. It's nothing special, just AUG. UAA, along with 2 other codons, is called stop codon. So UAA is one example of a stop codon. So how do we read this mRNA to make a protein? How does it work? So we need ribosomes to come in. So what are ribosomes? Real quick, I'll quickly show you what ribosomes are. So if here we have a cell, can you tell me where the ribosomes are on this picture? Here, this is a ribosome. That's one place. So this is one place where you can find a ribosome. And then here is another place on these little structures here. So this one that is free floating in the cytoplasm, it's not attached to anything. We call these what? We call them free ribosomes. Free ribosomes. And they are located free in the cytoplasm. These are ribosomes that will make proteins, and they will be used inside the cell. So whatever ribosomes, okay, just quick. Ribosomes are little structures that we can see here that are going to make this protein. It's like the machinery. It's like it represents it is represented by Bob the Builder in our story. Bob the Builder is the machine. It's the thing that's going to build this protein with its hands, right? So that's what ribosome. It's pretty much like the machine that's going to build this protein. So let me bring in some pieces. How does a ribosome look like? Before I continue what I was doing there. A ribosome has 2 units. A small subunit, this one, and then a large subunit. So essentially it's made up of 2 pieces. A small and a large subunit. This whole thing right here is called ribosome. We can give the top part the name large subunit because it's the larger one. And then we have the small subunit, the smaller one. Now, I said ribosomes can be either free, and whatever these free ribosomes, whenever they synthesize a protein, this protein will be used inside the cell. Whereas these right here, these ribosomes, are called bound ribosomes because they are bound to the rough endoplasmic reticulum. And whatever proteins these bound ribosomes make will be destined for outside of the cell. So let's say this ribosome makes a protein, that protein will be sent to another cell outside of this cell. Okay, so that's important to know. Now, what else do we need to know about this ribosome? I'll bring this picture in here for you to remember. This ribosome has 3 little spots here. 3 little spots. And you'll see why these spots matter shortly. We call this one here the exit spot or the E spot. And then we call this one here site 1 or the P spot. You don't need to know why it's called P spot. Okay, it's just remember it for now. And then the other side here is site 2, also called the A spot. How can we remember the P and the A and the E? That's easy. Think about it. A, P, E. It's a monkey. Ape. It's an ape. So that's how you can remember the other way of saying the 3 subunits. Now also, ribosomes are made up of proteins and rRNA. So that's funny. Ribosomes help make proteins, yet it is itself is made up of a protein. So that means you must use a ribosome to make a ribosome, because ribosomes make proteins, and we know ribosomes are made up of protein. So that's interesting, right? But besides proteins, they're also made up of rRNA. This is another kind of thing, just like mRNA. So it's another kind of DNA-like thing. So that's it for ribosomes that you need to know. So let's go back to this image. So now the first thing that happens in translation is we will have this, I'm going to delete this start codon here. But this small subunit is going to come in and bind to the mRNA, and then we will have, not yet, the large subunit will come in shortly. For now, it's not going to come in. The next thing that comes in is going to be something called a tRNA. A tRNA, and it looks like this. So what is tRNA? I'm going to quickly explain a little bit about tRNAs. So tRNA is called transfer RNA. Why? Because they carry amino acids. We know proteins, like I said before, let me go here. Proteins are made up of amino acids. So the amino acids can be considered as the materials to build the protein. So if we go back here, the tRNA carries or transfers these amino acids together to form a protein. So you can consider tRNAs to be like the material in the Bob the Builder story. Okay, that's what you can consider these tRNAs to be. So this whole thing here is simply made to carry this amino acid. So what does this look like? This reminds me a little bit of this plant. Remember this one? I forgot the name just now, but I forgot a clover. I think it's a clover. Anyways, yeah, it looks a lot like this, right? The stem is this part carrying the amino acid at the end. Anyways, doesn't matter. Now, notice here, the one end, the stem end, carries the amino acid, and the other end has something called an anticodon. What is that? So remember we learned here, let me go back here. We learned that these right here, every 3 letters is a codon. Now, this right here, this part of the amino acid is the anticodon. So it has the complementary code to this codon. So whatever this codon is, the specific amino acid that belongs to this codon will come and bring its anticodon to that codon along with the amino acid that it carries. So indirectly, this code codes for this specific amino acid, right? Do you see what I mean? Anyways, we're going to go back and come back just now. So this tRNA is made up of nucleotides. So you can see here, I started it. We have letter, 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 letter. It continues on letter, 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 all the way around. I just didn't fill it in because it would look like a mess. But just know that the rest of the tRNA molecule is continuously made up of these nucleotides. Now, the section where the amino acid binds always has ACC. You need to remember that as well. Okay, let's go back to our process here. So this specific start codon coded for a specific amino acid, a specific tRNA, it comes in along with its specific amino acid. So that's something else important to mention. Each tRNA carries its own unique amino acid, right? So we know there's like 20 different amino acid types. Each tRNA will carry the specific one that it codes, that its code is asking for, right? So anyways, now this tRNA comes in, it binds to the start codon, which is AUG always. Now, this big subunit is going to come in and bind to the whole thing. So now it's all one huge thing. And this, by the way, this whole step that I just explained is called initiation. It's the first step of translation, initiation. So what's the next step? So next, we're going to have the next tRNA come in, because we have now here a code, right? Oh yeah, remember this. Remember, this was the exit site. This was site 1. This one is site 2. So at any given moment, only 2 tRNA molecules can bind to a ribosome. So right now we have 1. So now here's another code, another codon that's going to ask for a specific amino acid. So now here comes another amino acid. Okay, the one that belongs to this codon. So it comes and it brings its own uniques because see, this code is different from this code. So this code is going to bring a different amino acid, the specific amino acid that corresponds to this code. So now it's going to bring it in, it's going to bind to site 2 by complementary base pairing. You can see, always matches, U to A to U, C to G, G to C, and so on, right? So now it binds, and now what happens is there's going to be a bond that's going to form between these two amino acids. We're going to start making a chain here. So it binds and forms this chain. And then as soon as this bond happens here, this tRNA, let me show you, this one is going to break off from its amino acid. So essentially, this amino acid was transferred to this tRNA here. And so this whole process right here is called elongation. So after this peptide bond is formed, we elongated this amino acid chain. It's basically, it became a bit longer. So this is called elongation. So first we had initiation, now we have elongation. So now the next step is going to be this. We're going to have this ribosome shift along now, shift along. So it's going to shift along because we're reading this code. We got to get all the way through it. So it shifts along. Now we have this amino acid that has no we have this tRNA that lost its amino acid that's going to exit with the exit side. So it's going to leave. So I'm going to delete it. It goes out, it leaves, and now this tRNA is going to go pick up the amino acid that it belongs to, right? It's going to pick up another one so that it can help build other proteins somewhere else. So let me show you. So here we have this process. So there's an enzyme called an activating enzyme, and it's going to take these tRNAs that are without amino acids, and it's going to give them their amino acid. So this one will come in, it will bind, and then it will take whatever the right amino acid is that belongs to this specific code here, and it will put it on it, okay? And this will form what we call charged tRNAs. Charged tRNAs. They can now help and go build other proteins. So right, so this happens now. So we elongated it, this one popped off. So now we're ready again at site 2 to receive another tRNA. So by the way, why do these codes, how do we know what type of amino acid these codes correspond to? So there's a little table that you need to know about, and it looks like this. So you can see that there are 3 bases: first base, second base, and third base. So we can see here, so for example, if we go back to our picture, let me go here. So let's say the first code here, AUG, let's go see what amino acid AUG corresponds to. So if we look, first base was A, so we're going to be in this column for sure. Now the second base is U, so second base is U, so it's going to be here, so we're going to be in this part. Third base is G, which is third base is G, so it's here. It's AUG. So we can see AUG is the start codon. It brings the enzyme, the amino acid called methionine. So you can see we are easily able to use this table to see exactly what amino acids this specific mRNA codes for. So we can go here and read all of these codons and see what amino acids will be formed, what chain of amino acids will be formed. So just bear in mind that table exists. Okay, so now we continue. So now again, we're going to elongate it even more. So elongation. So we're going to have another tRNA come in, bind by complementary base pairing. Now what's going to happen? Another bond is going to form here. This one in site 1 will detach from its amino acid. So now we have an elongated protein, right? We have now 3 amino acids. So guess what happens next? Next we have translocation, right? Translocation means movement. So now this ribosome is going to translocate, move to the new site. So now this one here without the amino acid is going to detach, leave, pick up its another amino acid. And now again, we have this empty spot to cause elongation to happen once more. So now here comes elongation. Another tRNA, it binds by complementary base pairing. What do we have now? We have another bond formed. So now elongation again. This one detaches. So now we have, this chain is getting longer and longer. We're making this protein according to this mRNA code. So now again, translocation. This ribosome will shift a little bit more. We're almost at the end of this chain. So here we go again. Now this one is in the exit site, so it's going to be forced to exit, pick up amino acid. Now we have another one here coming. And guess what happens now? What happens? Another bond forms. Another bond. And as it forms, what happens? This one will detach. So the whole chain is on this one in site 2. Now again, translocation happens one more time. So we translocate, we're near the end now. Notice we got now to the stop codon. So it's going to be interesting now to see how this process stops, why it doesn't keep going. So now this one again is in the exit site, it's going to leave, pick up its amino acid. Now we have this chain. How are we going to get this chain off of this tRNA so that the protein can be floating by itself? So now we reached this final stop codon, right? UAA is the stop codon. So how does that end? Basically, UAA codes for a tRNA that doesn't have an amino acid. So it's impossible to elongate this chain. Let's see how. So now if there's no amino acid, no bond can be formed. And we know that at this point, this one will detach, right? Because normally a bond will form and this one will detach. Now no bond forms and this one detaches. And this means that now the translocation will happen again. So now we'll move on a little bit more. So we're moving on. But now, guess what? When we move on and this one exits, now this chain is by itself. We made this protein be free. We have now a free protein. And once this happens, and there's nothing more to add, no more code, then this whole thing will just disintegrate. The big subunit and small subunit will separate. The mRNA will go somewhere else. So everything will just disintegrate. So this is how we synthesize this protein. So just to clarify real quick again, so the ribosome behaves like Bob the Builder. He is the thing that is going to build this protein. tRNA behaves like the materials. It brings the amino acids. And remember, mRNA here behaves as the paper, the little code that causes this protein to be made in this way. And this protein is what? This protein behaves as our rocket ship, the final target, right? The final target. So coming back to here. Now it makes more sense, right? Now it makes sense, this whole process. So real quick, I have a nice summary for you, something that makes translation look a bit better. So let me show you here. So ultimately, we made now this protein. This protein is able to stop his blood from bleeding, and he can now clot, and he can not lose so much blood. So that's one real example of how transcription and translation is used in our bodies. So here's a summary of translation just for you. So here we have initiation. Small subunit comes in, binds to mRNA. tRNA comes in for the start codon. Large subunit comes in, that's initiation. Next, we have elongation. A new tRNA comes in. They form a peptide bond. It detaches. This one at site 2. This is elongation. Now, next we have translocation. So this one that's so the ribosome will move along. This one will now be in the exit site. It will leave, and we have a new one coming in. So, and then we have termination. When we reach the end of the chain, everything will disintegrate and detach, and we will have a free polypeptide chain. So that's it. These are the 4 steps of translation. So we learned now about transcription and then the 4 steps of translation. So now to end it off, I want to quickly read you 2 definitions and then give you a past paper question so you can see and you can do it by yourself and see how they want you to answer it. So transcription is the process of copying a segment of DNA into mRNA. Now translation, the process by which the information carried by mRNA is decoded and used to build the sequence of amino acid that eventually forms a protein molecule. So hopefully these definitions make a bit more sense. So here we have IB questions and answers. So here is an example of a transcription question. I've noticed going through the past papers that they will never, thus far, they have never asked the question, explain transcription and translation in one question. That never happened. They always ask you to either explain transcription or either explain translation because there's too much information. It'd be impossible to have one question that asks you to do both in detail, right? But it can still happen in your here. So don't be surprised if you see it. Just prepare for both. So here's an example. You can read it by yourself and use my pictures and diagrams to see if it makes as much sense as possible. So this is one for transcription. Prokaryotes, by the way, transcription is the same in prokaryotes and eukaryotes. So don't worry, just prokaryotes is just trying to throw you off, but it's the same process that I just explained. And then here is explain the process of translation, and here's the answer. By the way, I want to note something. Notice how many lines there is. Each line here represents 1 point. All you need is 8 points. So if you say 8 of any of these things, there are so many, you can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. There are 13 things you can say. So if you just say 8 of these things, you'll get full marks. You don't need to say all of these, just 8 of them. Okay, so bear that in mind. So same with translation. There are so many points you can get. You only need to list 8 of them and you can get the full marks. Okay, so don't stress out too much. You don't need to know all of these. So I hope this was useful for you. This is all you need to know, all the details about transcription and translation.